So we're going to begin chapter 8 in the Pierce text, and just to make sure everybody is on the same page, we did the introductory chapters, chapter 1 and chapter 2, the first 2 weeks, and now we're jumping to chapter 8, and we're starting the molecular genetics portion of this, or of the class. So I have found over many semesters now that tackling this harder to master molecular genetics section is easier done in the beginning of the semester rather than saving it till the middle or the end of the semester. So chapters 8, 10, 12, 13, and 15 will be molecular genetics. All right, so because we are moving straight into molecular genetics, what we're really doing is we're starting to narrow down the size of the genetic material that we're dealing with. So chapter 2 really dealt with the whole entire cell and how we replicated genetic material within a cell. And if you guys remember from the pyramid or the upside down pyramid diagram that we did the second activity of week 1, the next major section is the chromosome. So the chromosome is going to reside within the nuclear membrane of the cell. And so this chapter is all about chromosomes, their morphology, their anatomy, and a lot of the annotations and problems that we see when chromosomes copy. So to start this chapter, I just want to let you know a little bit about an ag example that has to do with chromosomes, and this is Blue Moon. And Blue Moon is a mule that was really quite an interesting genetic case. So most mules end up being sterile because they are a cross between a horse and a donkey. So a horse has 64 chromosomes and a donkey has 62 chromosomes. So when these individuals cross, usually the mules end up with 63 chromosomes, and the one that is lost is the one that doesn't enable reproduction to take place. So that this is just a visualization of how this takes place. So we end up with either a mule if it's a male, or a henny if it's a female, but realize that they are not fertile individuals if they're a cross between the horse and the donkey. So normally this is a general rule of thumb, but as with lots of things that we deal with, there was an exception to the sterility rule. And this happened in 1985 with a female mule whose name was Krause. And she ended up being pastured with a male donkey and was obviously bred and then had a baby named Blue Moon. And so there were no other females in the pasture, and it was just these two individuals. And so because this was such a conundrum relative to what had already been shown many, many, many times as far as mules being sterile, the veterinarian and the producer took blood samples, sent it off for DNA testing, and then they genotyped 17 genetic markers within the genome. And so they were able to create a karyotype for Blue Moon, and who was the dam, and they ended up finding that she had 63 chromosomes, which is a normal amount of chromosomes for a mule. But what ended up happening is that she, instead of being sterile, she ended up being fertile. So what ended up happening is Blue Moon inherited the entire set of horse chromosomes from her mom, and no segregation took place. So the chance of that happening is really once in a blue moon. That's why she got her name. So what ended up happening is Blue Moon then had Krause because she was fertile, and so it gets a little bit gray, but Krause and Blue Moon essentially are out of the same sire, and they are also mother and son, but in kind of an odd way, they're also brother and sister because they're out of the same sire. So that gets a little bit or it gets to be a little bit of a conundrum as well. But what ended up happening is when the chromosomes lined up on the metaphyseal plate, they ended up exactly in order so that all of the chromosomes from the dam and all of the chromosomes from the sire lined up perfectly in line with one another. Whereas typical meiosis looks something like this. So we get, a copy from mom, copy from dad, copy from mom, copy from dad, and they're very, very random. So the chance of meiosis in Krause happening was that alignment was very, very small. So this is how Krause ended up being fertile, and really leads us into the variation that we see associated with chromosomes in different organisms. So most species are going to have the same number of chromosomes. All of those chromosomes are going to have a very distinct size and structure. However, variations do occur, and these variations tend to be chromosomal mutations, and those mutations can result in variations either in the number or the structure of chromosomes. And we're going to look in depth at both of those situations. And this chromosomal mutation isn't necessarily always a bad thing. In fact, it's really played an important role in evolution and how we've been able to adapt over time to new environmental stressors. So just to review, a functional chromosome is going to have two major types of anatomy. They're going to have a centromere, which you see right here in the center, and then they're going to have 4 telomeres. And those telomeres are going to be the ends of the chromosomes, and they're going to help stabilize the genetic material that we see on those ends. The chromosome can then be split up into 4 basic types of morphology. So the one that we're used to looking at is the metacentric morphology. That's where the centrosome or centromere is going to be right in the center of the chromosome. However, that centromere could be located more towards one end of the chromosome compared to the other. That configuration is submetacentric. If you get a chromosome actually appearing very close to the end of the chromosomes, that's going to be acrocentric. And then finally, when the chromosome comes together and is held together at the very ends of the telomeres, that's going to be telocentric. So again, telocentric is the easiest one to remember because the chromosome is actually holding the chromosomes together by the telomeres. So in addition, as these different morphologies occur, so submetacentric, acrocentric, telocentric, the short arm is going to be referred to as the P arm, and the long arm is going to be referred to as Q. So just put that in your notes because that is going to become important as we start looking at some of the other mutations. So karyotype, we've talked a little bit about this, is going to be a set of chromosomes that is possessed by an individual organism. And the karyotype is how we visualize those chromosomes. Usually it's presented as a digital picture, and the genetic information is actually treated to be halted during metaphase. And the chromosomes are going to be organized by size. So chromosome number 1 always has the most genetic material, and then the last chromosome in humans would be 22, is going to have littler amounts of genetic material. So in order to prepare or create a karyotype in a lab, what ends up taking place is a sample will be taken from an individual organism, whether that's a plant, an animal, or a human. And those samples need to be taken from an actively dividing cell. So bone marrow, white blood cells, or meristematic tissue in plants. That tissue sample is then going to be treated with colchicine, and this is going to prevent the full process of the M phase, mitosis and meiosis, from taking place, so that the genetic material is halted in metaphase and cannot proceed to anaphase. And then at this point, the cells are going to be spread on the slide, stained, and photographed. And staining is going to happen one of 4 ways. Those 4 ways are shown here on this slide. We can stain for the G banding configuration, which stains the AT base pairs. We can stain for Q banding, which is going to be a mustard stain that it can be visualized with UV light. We can also stain with C banding, where we actually dye or stain the centromere region. And then we can also stain with R banding, which looks at the guanine cytosine base pairing. And so one entire sample will be treated for these 4 different types of stains, and then they'll be visualized digitally, and we're going to be able to match up chromosome 1 with chromosome 1, chromosome 2 with chromosome 2, based on what we know about how the genetic information is actually configured in the genome of that organism. So this gives you an introduction to Chapter 8, and this is going to be part A, so don't forget to watch the other parts of